Hey everyone, and welcome to our lecture series on the reproductive system. This is actually going to tie in with the final chapter for this unit where we talk about development in pregnancy. So before we can get into all that cool stuff, we need to talk about the reproductive anatomy and physiology of the males and females. Now, this is a 3-video lecture series for the reproductive system. Today we're going to go through our male reproductive system, looking at both the structure of all of the male bits and bobs, as well as physiology behind things like spermatogenesis. And then what we've done is the last 2 videos are going to cover the female reproductive anatomy and then female reproductive physiology, because it gets a little complex with the hormones and the eggs and all that stuff, and I didn't want to overwhelm you with a very, very long video. So let's get started. Now, reproduction in general is weird. Across all species, it's kind of weird how we get a sperm to an egg. There are species that just undergo what's called parthenogenesis, where there's no need for sperm. The insect world is rather weird. You have basically beetles that will take what is essentially a phallus and just stab it into the abdomen of a female. You have different dragonflies that actually have deposit areas for different sperms, so they can mate with different males and then choose which sperm they like. The males have then evolved with like little scrapers to scrape out competing sperm. And then one of my favorites is the anglerfish, which lives deep in the sea. The anglerfish, in this case, the female is the larger of the 2 sexes. And when copulation occurs, the male actually physically attaches itself to the female and disintegrates and basically becomes a sac of sperm that she can use when she's ready to create her eggs. So it's kind of weird. It's wild. I have a video here that's also very funny. I encourage you to watch it. It's quite good. So what we're focused on, obviously, is the human aspect of reproduction. So in this video today, we're going to talk about the male reproductive anatomy and physiology, as I mentioned. Then we'll go through the female, and then in our next chapter, we're going to talk about what happens if the egg and sperm get together. So we'll kind of see what happens there. So here is a quick overview of the male reproductive system. This is a mid-sagittal view. This is basically the same view you're going to see on pretty much all the models in the lab room. It shows a lot of different structures here that we'll be going through, and also some structures that we're not going to be going through. But for our purposes, we're actually going to start where everything essentially begins in terms of sperm production, which is in the scrotum and testes, and we'll kind of make our way out through the penis. So that's kind of the route we're taking. So the scrotum is the sort of sac outside the body in which the testes, epididymis, and our spermatic cord are going to exist. Now, if you were to open up the scrotal sac, there are 3 things that you're going to see. So first and foremost, there is actually a septum, a little ridge of tissue on the internal aspect of the scrotum. It actually lines up with what's called the raphe or the raphe, which is that outer line that you see on the scrotum that runs midline. So you can kind of tell where the right and left testis is. So this scrotal septum is there to help ensure that the testes kind of stay in their own lane. So one on the right, one on the left. The other thing that I want to mention about the scrotum is that it contains 2 muscles, the cremaster and the dartus muscle. Why? Well, that kind of brings up the topic of why the heck the testes are outside of the body. As we'll see with females, our gonads, the site where we produce eggs, is all internal. Well, as it turns out, in order to produce sperm properly, we cannot keep them in the abdomen. It's too hot. Sperm actually have a lower temperature in the low 90s that is preferable for development. So if we kept them in the abdomen, we would not have sperm development. So by keeping them outside the body, we can regulate their temperature a lot easier. Using the dartus and cremaster muscles, we can adjust how close the testes are to the body, and we can adjust how much surface area the scrotal sac has to radiate heat off or keep heat in. So the cremaster muscle, which is this deeper muscle that you see here, It's actually an extension of your internal oblique muscle, and it kind of comes up through that inguinal canal and wraps around the testis and epididymis. So when it contracts, it's going to lift the testis up. Now, the dartus muscle we can see is this sort of white line here. It's the dartus fascia. It's going to go around the outside of that cremaster muscle. And it's actually connected to the scrotal skin, and what it's doing is not the lifting, but it actually causes the scrotal skin to wrinkle. So when it contracts, it's going to wrinkle the skin, and that's going to affect surface area. So when the testes get too hot, what happens is the cremaster muscle will relax and the testes will fall away from the core. So they'll hang lower so that they are not as hot as body temperature. Then the dartus muscle is going to also relax, and that's going to cause the scrotal skin to also not be wrinkled. So it increases surface area, and the more surface area I have of my skin, the more sort of evaporative cooling that I have. So basically, when it's hot, both muscles will relax, increasing surface area for evaporative cooling, and then also pulling the testes away from the body to keep them cooler. And then when it's cold, the reverse happens. So now it's too cold, the cremaster muscle is going to contract. It's going to pull the testes up closer to the core where it's warmer. And that dartus muscle is going to contract and wrinkle the skin on the scrotum. That's going to reduce the surface area, and that's going to decrease heat loss. Okay, so that's what's going on when your testes are changing their position due to temperature. So here is a testis. This is an actual cross-section of both a testis, which is the oval gland, and then outside of it is the epididymis, which is something we'll talk about later. The testes are actually surrounded by two layers, tunicas, that have a couple different roles. So the first one is the tunica vaginalis, which is right here. And you can actually see that the tunica vaginalis has a very similar structure to, say, the pericardium. It's a dual layered membrane. One layer is on like the scrotal sac, one layer is on the testis itself. There is a small space in there with a very, very thin amount of fluid. It's more of what we'd like to call a potential space, where, you know, it has the potential to hold stuff, but in reality, we don't really put anything in there. It just gives the testes somewhere to move. Clinically, this is a significant space because we're going to talk about a condition later on that's called a hydrocoele. And in patients that have hydrocoeles, this is where the fluid is building up. So it's not in the testis itself, it's in the space between the two layers of the tunica vaginalis. Now, deep to that visceral tunica vaginalis, we have a thicker tissue called the tunica albuginea. This is dense connective tissue, and it's important for a couple different reasons. First, as you can see, it's going to go into the testis, and it creates these spaces for the seminiferous tubules to exist. We call these lobules. The other thing that we found out about the tunica albuginea within the last 20 years is that we are pretty sure that it is involved in the motility of sperm. There was a study done in like 2002 that showed that there are contractile elements in the tunica albuginea, especially near that ready testis, which is right here in the middle, that are doing these random contractions. And we think that might be important for moving the sperm through the tubules. And because it's a thicker tissue, it's also responsible for maintaining the structure of the testis as well. Now, within each of these lobules, we have these really coiled tubes that we call the seminiferous tubules. These are important. The seminiferous tubules are where we're going to have our first physiological process of this chapter called spermatogenesis, which is the creation of sperm. So in males, our gonads are the testes and the gametes are sperm. So the sperm are created within the seminiferous tubules through the process of spermatogenesis. And they're going to go through another series of tubules. We have straight tubules right here to the ready testis, which looks like a little net. And essentially the ready testis is the connecting point between the testes and the epididymis. So it's like the next step on the way out as we're heading up into the spermatic cord. Now, if we take a look at the seminiferous tubules, we're going to find three populations of cells. The first population is a series of stem cells, so that means they can basically generate into anything. They're called spermatogonia. And as we will see, the spermatogonia, the stem cells particularly, in general, let's just say stem cells in general, of males are different than female. So as we'll see with the female, we have stem cells, but they divide and they start to become primary oocytes. In males, as we will soon see, stem cells will undergo mitosis. One half will remain a stem cell. The other half will continue on to become a sperm cell. This is why males are able to produce gametes basically throughout their entire life. Now, the other two populations of cells we have, these are here for support. So the sustentacular or Sertoli cells, these are important because they're kind of the nursemaids for the sperm. They're going to make sure the developing sperm have all the nutrients they need, and they are the ones to let them go into the lumen once they've reached the right stage of development. The Sertoli cells are also going to create what's called the blood-testis barrier. We're going to see a lot of blood blank barriers in this class. Basically, what these Sertoli cells are doing is they are masking the sperm from the body's immune system so that there isn't an immune reaction. They are undergoing meiosis, so they're going to look a little bit different with the chromosomal arrangement. And so this just kind of helps prevent the body's immune system from attacking its own sperm cells. And then outside of these seminiferous tubules, we have sort of these interstitial cells, which are called Leydig cells. And what their job is to secrete testosterone. So testosterone is critical for the development of sperm. You could have all the parts, but if you don't have adequate levels of testosterone, it's going to affect your ability to produce sperm. And you're going to go into all that in a lot more detail when you get into physiology. So let's talk about how we make sperm. Again, we're going to be using a lot of different staging here. So you can see there's lots of bolded terms. Essentially, if you think all the way back to our first unit when we talked about cell division, mitosis, we also talked about the process of meiosis, which is how we create haploid cells, half as much genetic material. Spermatogenesis is meiosis perfectly, okay? So we start off with our spermatogonia. They are diploid. They have a full set of chromosomes. One of those spermatogonia will undergo mitosis, and one of those daughter cells becomes a primary spermatocyte. We have not reduced chromosome number. We're still diploid. They'll undergo meiosis 1 and go through that process of reducing their genetic number, becoming a secondary spermatocyte. The secondary spermatocyte undergoes meiosis 2, becomes a spermatid, and then the spermatids are going to undergo something called spermiogenesis, which is essentially where they mature and get all the bits and bobs so they actually look like a sperm. And now they are ready to go and proceed through the rest of the tubules. So this is what it looks like. We start off with our spermatogonia that's going to undergo mitosis. One of the daughter cells stays as a spermatogonium. It remains as a stem cell, so you can continue, basically perpetually produce sperm for your life. There is a small decrease as males get older just because of natural testosterone decline, but overall they can produce sperm forever. The other daughter cell is going to become a primary spermatocyte. That primary spermatocyte undergoes meiosis 1, which remember the main function for meiosis 1 is we're going to increase genetic variability. We have crossing over and all that fun stuff in prophase 1. And we're reducing our genetic number. We go from 46 to 23 chromosomes essentially. Now, at the end of meiosis 1, we have 2 secondary spermatocytes. These cells are haploid, and they're going to go through meiosis 2, which if you remember is just mitosis. We're going to go through all the phases of mitosis, make sure we have all our chromosomes present. And we end up with 4 spermatids. Now, this diagram isn't completely accurate. The spermatids are actually physically connected to one another. at this stage. And they're going to go through that process of spermatogenesis where we actually create the sperm. And we'll take a look at sperm in just a minute, but functional sperm need to have a tail for locomotion, and they need to have a midpiece for energy, and they need to have an acrosome on the head. That acrosome is important because it's going to provide the enzymes needed to penetrate a thick layer around the egg. And if we don't have that, we can't get through that, we cannot fertilize. So here's a sperm, okay? At the head of the sperm, this is where the nucleus is, so that's where the genetic material is located. And you can see there's this sac at the head of the sperm. This is enzymes. This is what's going to break down that zona pellucida, and basically whoever can break it down faster and touch the egg's membrane first is the winner. In order to do that, they have to move through the female reproductive tract, okay, which is perilous at best. And we'll go through all those reasons when we get to the female reproductive system. So they need to have a tail that is used for locomotion. Okay, so the tail is going to spin, and then we have the mid piece, which is where all the energy is coming from. There's lots of different things that can go wrong here that can affect fertility. So if we can't produce an acrosome, can't get through the zona pellucida. If we don't have a properly functioning tail or enough energy, you're not going to have the ability to move through the female reproductive tract. These guys also don't live for a long time. Most of them, keyword here being most, are going to die within 2 days, 48 hours in the female reproductive tract. Some can live as long as 7 days. So when we start talking about the female cycle, knowing when you ovulate is important, but there needs to be other means there if you're trying to prevent pregnancy, because these guys can last for a long time. The egg has about a 24-hour window of viability. So if there was an ejaculate prior to ovulation or shortly after ovulation, you are in the realm of getting pregnant. Now, we've made our sperm in the testes. They now need to make their way to this comma-shaped structure on the sort of posterior lateral side that we call the epididymis. This guy right here, okay? In order to do that, they go through the seminiferous tubules, into the straight tubules, into that ready testis, and from there, they enter the epididymis. Now, the epididymis is actually just a big old tube. It's about 20 feet long in that small organ, and this is where sperm are maturing. So they're continuing that process of getting everything that they need so they can be successful at fertilization. Now, the tube that's inside the epididymis, so the epididymis is the organ, the tube inside of it is called the ductus epididymis. It's a larger lumen, and it's lined with cells that are called stereocilia. And the stereocilia are there because some sperm don't make it, and they don't last forever, so they're just reabsorbed in the body. And we also store sperm in the epididymis. We also store it in this tube that's going to go up from the epididymis that we call the ductus or the vas deferens. The vas deferens is a tube that's going to take sperm from outside the body in the scrotum to the accessory glands within the pelvic cavity. Okay, so it's a transport tube, and its other job is to store sperm. Okay, so this is important because it can store sperm for a long time. We're going to talk about a procedure shortly. I need you to keep that fact in mind. Now, one thing that I hope you guys have been asking yourselves is if the epididymis, the vas deferens, the testes, they're all outside the body, where are they getting their blood supply from? They're getting it through, obviously, arteries and veins. We also have nerves that need to go down there to convey information. But how do they get there? So there is this opening within the abdominal cavity. We call it an inguinal ring. And it creates like a tunnel, a legitimate tunnel through your abdominal wall muscles right at sort of like the V line, just above the genitals. And this is called your inguinal canal. It's in that inguinal region. This is how the arteries and veins, the lymphatic vessels, the nerves, and that vas deferens are going to go from the outside of the body to the inside. So we bundle all those tubes together and we create what's called the spermatic cord. And it's going to come up through that inguinal canal. And the inguinal canal is going to be important at the end of this lecture when we talk about development as well, because that's actually how the testes get out of the abdomen. So speaking of spermatic cord, there is a procedure that males can have done called a vasectomy, which is essentially where they cut off the ability for sperm to be present in semen. So they can have an ejaculation without semen, without sperm, which would again basically make it so they can't get someone pregnant. Is it 100% foolproof? No. And this is where understanding how the vas deferens work in the male reproductive tract is really important. So how vasectomies are done, they can be done a couple ways. It's outpatient. They make a small incision within the scrotum. They will find the vas deferens and they can either cauterize it with heat, or they can do what's called a cut and ligation, where they cut it and they tie the ends together. So ideally what would happen is you're still producing sperm, but it's just getting reabsorbed into the body. And now when you have an ejaculation, you'll still have semen because all of that fluid is coming from accessory organs. It just won't have sperm in it, okay? The thing is, you have to make sure you wait a certain amount of time and clear the pipes, as it were, because you could still get someone pregnant after having a vasectomy for two reasons. One, you didn't wait long enough and there were still sperm within the vas deferens that were present. And two, unfortunately, sometimes these ends can actually regrow. So that can also happen after a vasectomy. So there you are. Now, what are these accessory glands that I've been talking about? There are 3 accessory glands that are on the inside of the male in the pelvic cavity, and their job is to basically produce this fluid through which the sperm can be ejaculated into the female and have everything it needs to get to the egg. So we have our seminal vesicles, which are going to be alkaline, which remember means they're going to have a high pH, and they're also going to have sugar and prostaglandins. So why do they have that? The female reproductive tract, as I mentioned, is treacherous for sperm. It's highly acidic. We typically only ovulate on one side a month, so we alternate right and left typically. So they can make a wrong turn, they have to get through the cervix, there's a lot of nooks and crannies they can get caught up in, the cilia can trap them, there's a lot of ways this won't go right. So one of the protective mechanisms the males have evolved is they send the sperm into the female reproductive tract surrounded in an alkaline fluid to off-put the acidic environment of basically the vagina. The fructose is there as sugar because we need sugar to convert into energy with the mitochondria so that they can swim. And prostaglandins, we'll talk a little bit more about those. We've talked about them briefly when we discussed oxytocin, but they cause smooth muscle contractions. So they can actually help the female reproductive tract contract and help kind of push the sperm up a little bit. And this makes up most of the volume of semen. The other accessory gland we have is the prostate. This one has a little bit of acid in it, just to kind of help balance out pH, and again, a bunch of other stuff that we're not super concerned with in our class, but it's there to help with the sperm. And then the last gland we have is about the size of your pinky nail, right in the pelvic diaphragm, called the bulbourethral or Cowper gland. And this has 2 functions. The first one is, again, the male's reproductive tract is shared with the urinary tract. Urine is acidic by nature. Sperm does not like acid environments. So prior to ejaculation, the bulbourethral gland will secrete a mucus that will go through the urethra that essentially neutralizes any leftover urine present and changes the pH there. It's also going to lubricate the tip of the penis, which will help with penetration during copulation. So let's talk about some of these ducts before we get, or these organs before we get into the rest of the tubes. So here is your vas deferens. It actually comes from the scrotum, and it comes from the front and wraps around the back. So it kind of goes like this around the back of the bladder, and it meets up with the seminal vesicle here. This is your prostate gland. So the prostate actually lies right underneath the bladder, and the urethra goes right through vertically through the prostate gland. This is why males that have prostate gland issues, whether it's benign hyperplasia or maybe it's a malignancy with prostate cancer, they can usually tell there might be something wrong with the prostate just by how their urine flow is. It might be decreased in strength, it might have more spurts. That's one of the ways we can kind of recognize it initially without going through a palpation. Now, the other duct that we want to look at, here's our pelvic diaphragm. You can see these little guys here, right in the pelvic diaphragm. Those are your bulbourethral glands. Now, they are secreting fluid into the remaining duct system. There are two remaining tubes that are helping propel the sperm out of the body. The first is the ejaculatory duct. Now, the ejaculatory duct is formed when the seminal vesicle and the ampulla, which is the widened portion of the ductus deferens, meet together. And it actually, when you look at a sagittal section of the prostate, you're going to see a vertical tube. This is going to the bladder. This is the urethra. Then you're going to see a tube at an angle. This one is the ejaculatory duct. It's connecting to the seminal vesicle and vas deferens posteriorly. So the angled tube, that's the ejaculatory duct. That's where the seminal fluid's going to come in from. That's where the sperm's going to come in from. And then you have the prostatic urethra. Okay? From there, we go into the membranous and the penile urethra, which then exits at the external urethral orifice. And now the sperm is out of the body. What is semen? Semen, as I mentioned, is a mix of sperm and all of this seminal fluid, prostatic fluid, etc. It is anywhere between 2.5 to 5 mils of ejaculate with 50 to 150 million sperm per milliliter. It's a lot. So the reproductive strategies of males and females is very different in that males produce a huge number of gametes because they have to go through, again, a very treacherous environment to get to the egg. So we need to have a lot of guys to get there, because if we only send one, the likelihood that they make it is very, very low, okay? But the egg isn't waiting, so they just release one. What's really interesting is after ejaculation, we really don't, and when I say we don't know why the function happens, we have an idea, but we're not, haven't proved it yet. But semen will actually coagulate, and then it will reliquefy. And so likely the reason is so that it will stay within the female reproductive tract, giving it time to get into that cervix, into the uterus. But again, we aren't entirely sure why this happens. So how do we get the sperm into the female reproductive tract? Through the penis. The penis is the copulatory organ. That's really all it is used for. Okay? It's just a way to get sperm into the female reproductive tract, more precisely to get it as close to the cervix as possible so that they don't have to travel as far. Now, the body of the penis, which is what you see primarily, it has two erectile tissues within it, the corpus cavernosum and the corpus spongiosum. So here's a sagittal view. The corpus cavernosum is going to be on the dorsal surface of the penis, right here. These larger cavernous structures here. Running through the center of each corpus cavernosum is a blood vessel. And what happens during an erection is that area will engorge with blood, and it will compress the veins that are in that area. So blood will continue to pump and fill these spaces within the corpus cavernosum, and it can't retreat out. So that's how you get the increase of rigidity and increase of size during an erection. Now, one thing that we have to keep in mind is, again, The penis is how we get the sperm out. So if that corpus cavernosum continues to enlarge and get rigid, it can actually compress the urethra, and now nothing can get out. So the urethra itself also has a set of erectile tissue that goes around it, which is right here. So you can see that little opening. That's the urethra. And then this is what we call the corpus spongiosum. It goes around the spongy urethra. It's going to harden as well, essentially, and keep the urethra open during ejaculation. Now, the penis is actually connected to the body here at the root, and the bulb, which is the big fat part right here, that's physically connected to muscles in your perineum. So that is how the penis is physically attached to the pelvic area. On the opposite end of the spectrum, we have the tip of the penis, which is the glans penis. It is covered by essentially extra skin that we call the prepuce, which is the foreskin, and it can be removed in a process called circumcision. That just keeps the glans penis exposed now. We're going to talk in the next video about the female reproductive system, and there's actually some structures like the glans penis, the prepuce, the scrotum that all have homologous structures within the female as well. So the last couple things I want to talk to you guys are related to development and some things that can arise in early infancy and childhood. Now, in our next chapter, we're going to go through the process of fetal development. We're going to talk about sex determination. But once sex has been determined, about week 10, the testes will start to develop and descend from the abdomen. So they start off in your abdomen, and then they'll start to descend down into the scrotum through the inguinal canal. And it takes up to 20 weeks for this to happen, okay? Now, about 3% of males born are born with what's called cryptorchidism, which means the testes are not descended fully into the scrotum. This is a diagram from UC Davis Pediatric Urology, and you can see that there are actually a ton of different placements that the testes can be in, okay? This is often picked up by the pediatrician. They'll be able to notice that there's not a testis there. It can be confirmed through ultrasound. This tends to occur more in premature infants because, again, especially if you're born before 35 weeks, you're still within the realm of possibility that they're on their way down. They just haven't completed that descent. One nice thing about this and the other disorder we'll talk about is that most children, if they are born with an undescended testis, it will descend by about 6 months of age. After that point, though, that's where you have to start thinking about surgery, because there are some complications that arise when you have testes that remain in the abdomen. So first and foremost, it increases your risk of infertility, because remember, testes are not meant to be in a warm environment like the abdomen, and you can't produce sperm at that area. The longer they are outside the scrotum, the more damage cellularly is actually occurring to the testis. So this also increases your risk for testicular cancer. It increases the risk for testicular torsion because we don't have that connective tissue, that tunica albuginea, the spermatic cord, the scrotal septum to help make sure that the testis doesn't twist within the scrotum. And that's one of the clinical applications that I've posted for you guys, is to kind of research what is testicular torsion, signs and symptoms, and how it's treated. But yeah, that's our first sort of developmental thing to talk about. The last one that I want to talk about is something that's also fairly common in male infants, which is called a hydrocoele. This can happen. It's very normal after birth to see fluid in the scrotum. Okay, so typically they'll shine a flashlight on it and you can see like this outline of light, and that's indicative of there being fluid in the scrotum. Again, it's very common right after birth because it takes some time for that inguinal canal area to seal itself off. Okay. Now, the longer that inguinal canal connection is open, it's open to your abdominal cavity. And fluid from the abdominal pelvic cavity can just drain into that space between the tunica vaginalis layers and fill with fluid. and that creates an enlarged, usually it's one-sided, unilateral side of the scrotum. It's painless, it does not hurt, but it's definitely a noticeable bulge, okay? A lot of times this can resolve itself after, maybe by the time they're a year, it can typically go away. Any fluid that's left can be reabsorbed, and they're usually fine. However, after a year, if it's still present, it can mean you need to have surgery to repair it. And there is a complication furthered by having this inguinal canal open for longer, is that with increased intra-abdominal pressure, like when the kid is pooping, it can actually force the bowel into the inguinal canal, basically creating an inguinal hernia. And that causes lots of problems because the bowel can become trapped and ischemic, and then we have a bigger problem on our hands. The surgery is very easy. They do a small incision, seal the hole up, and you're good to go. So that is one topic I wanted to discuss with you guys. I will see you in the next video when we go through the female reproductive anatomy.